Hello. In this lecture, we are going to be talking about the endocrine system. So I'm dividing this chapter up into two lectures. In the first lecture, I'm going to get an overview of the endocrine system and sort of what its function is in the body. In the next lecture, we're going to be looking at a lot of the different endocrine organs and the hormones that they secrete. So let's start off with the function. So the endocrine system shares the function of communication with the nervous system. However, how they communicate is a little bit differently from one another. So overall, both of them are going to be communicating ultimately to maintain homeostasis and respond to their environment. However, a lot of the other things are going to be somewhat different, although they'll share some things. So, in the case of the endocrine system, the signal is going to be chemical in nature. Essentially, you're going to release hormones into the bloodstream in the case of the endocrine system, while the nervous system, while it does make use of chemicals in the form of neurotransmitters, most of the signal that is being sent here is electrical as changes in membrane potential traveling down the neuron and the axon of that neuron. Because of the nature of their signal, the endocrine system is going to be fairly slow to respond compared to the nervous system. Essentially, the chemicals need to diffuse through the blood and diffuse through the body, while in the case of the nervous system, you have a very direct attachment to the communicator and the communicatee in that case. Uh, Although it's slow, it tends to linger because you have to remove the chemicals from the bloodstream, while the duration of the nervous signals is going to be relatively short because you're able to remove that signal very rapidly. The endocrine system is going to be less specific because it's sending its signal into the bloodstream and diffusing throughout the body. Any receptors that are going to be specific to the hormone are going to respond to it. So it's specific in the sense that you need the receptor there, but it's non-specific in that it's not going to a specific location. The nervous system, on the other hand, is more specific. The signal is directly going to a spot in the body, and so you are going to only be stimulating that spot. So, endocrine system, we call this kind of signaling endocrine in nature, while the signaling in the nervous system is called paracrine. And I'll explain what these two terms mean in the next slide. But for paracrine, in the case of the nervous system, this is sometimes seen as an even more specific signaling mechanism known as neuronal. All right, so what is the difference between endocrine signaling and paracrine signaling? Well, with endocrine signaling, you have the molecule being delivered via blood to another target cell. So the cell sending the signal is going to be farther away than the cell receiving the signal. Paracrine, on the other hand, is when the molecules are sent just a short distance to neighboring cells. So, like in the nervous system, you have the axon terminal interacting with the dendrite or the effector cell, and so it's very close in proximity, the cells that are interacting. You can also have another kind of signaling that's known as autocrine. That's where the cell that sends the signal also receives it. This is less of a function that we'll see in our chemical signaling discussions. It does play an important role in development and in other functions in the body, but we won't be talking about it in detail. recall when we talked about skin in the previous A&P class, that we talked about glands and how we had exocrine glands and endocrine glands. Well, the difference between these two glands has to do with where the secretion is going, and ultimately whether it is being in put into a duct of some sort. So endocrine signaling is ductless. It secretes the contents into the extracellular fluids, which are ultimately going to then diffuse into the blood. Exocrine signaling, on the other hand, has ducts. So you can see over here on the right, this has a ducts. These green cells are what are secreting the compound, the secretions in the case of the exocrine secretions, whereas these cells over here on the endocrine, they're secreting it, but they're not going to the duct, they're going into this extracellular fluid, and the extracellular fluid ultimately is going to go into the blood. So the blood, in this case, exocrine, it's going out like this, whereas this just goes kind of everywhere in all different locations, but will ultimately end up going into the capillaries and moving then through the bloodstream. There are two major ways that we two major groups that we can classify hormones. One is going to be the water-soluble hormones. The other is going to be the lipid-soluble hormones. So over here on the right, we have those that are lipid-soluble. These are generally going to be cholesterol derivatives, and how they signal is going to be slightly different than the hormones that are over here, which are going to be the water soluble ones. So water soluble, these generally are derived from amino acids, comparing this to the lipid soluble ones that are generally derived from cholesterol. Now, the water-soluble ones we actually can break up into more categories. Some of them are going to be short peptides, others are going to be derivatives of the amino acids, like the catecholamines, which are derived from tyrosine. But for this case, just know that water-soluble ones are going to be derived from amino acids of some sort. Lipid ones are going to be derived from cholesterol. The significance of this is in how they are signaling. So in the case of the water-soluble ones, you don't actually go into the cell, but instead you bind to what's known as a receptor on the surface of the cell. And this leads to a signaling pathway that ultimately is going to have a number of different functions. Usually it's going to change the way that a protein is going to interact. So you can see here, you have the receptor, binding of it leads to activation of a effector protein, which is going to produce a second messenger like cyclic AMP. This then activates another protein like a kinase. The kinase can then phosphorylate it. And this is just one way that receptor-bound proteins can interact, or receptor hormones can interact. There are other signaling pathways, but the key here is that it's acting on a receptor on the surface of the cell and generally leads to changes in the protein. So these tend to be shorter lasting because you just remove the receptor or the ligand from the receptor, and it will end the signaling pathway. Lipid signaling, on the other hand, so you have this lipid. Now, it's not water soluble, so it's actually going to be bound to a protein in the cell. So you have like your albumin and other proteins. It's bound to that in the blood, and then it diffuses out of the capillary into the extracellular fluid. Um, Now, because it's lipid soluble, it actually is able to go through the plasma membrane, and here it'll bind to a receptor. Now, this receptor is actually going to be an intracellular receptor because it's inside the cell. And once it binds to it, it's going to move into the nucleus, bind to the DNA, and ultimately lead to a change in gene expression. So you have the DNA leading to the synthesis of the mRNA down here. The mRNA goes out, you have the ribosomes synthesizing the protein. And so this will tend to be slower to react, but longer lasting. Because the change in gene expression is more of a long-term response. All right, so what determines the effect that a hormone will have? So first of all, we have the type of hormone that is going to be bound. So not all hormones are created equal. So we have some hormones like norepinephrine, and even a similar one like epinephrine, that are going to have different responses because they're different hormones. And these are similar, there's some overlap in their responses. A better example would be like norepinephrine compared to insulin. These are completely different hormones. They're not related in any way except that they're both water soluble, so they bind to receptors on the cell membrane. But insulin is a peptide, while norepinephrine is an amino acid derivative. So the type of hormone matters. The second thing that can affect it is the type of receptor. So over here on the left, we have an alpha adrenergic receptor. Over here, we have a beta adrenergic receptor. Norepinephrine and epinephrine can bind to both of these receptors, but they do so with different affinities. And so that means that it's going to be more likely to bind one versus the other. And so the type of receptor that's present can lead to a change. You can also have differences in the second messenger response. So there's several different second messengers that can be produced. One is the cyclic AMP that we saw earlier. IP3 is another second messenger that could be produced. So over here, you might have cyclic AMP. That should be a lowercase c. Yeah, it's not obvious with how I drew it. Well, over here, the second messenger might be IP3, and those can lead to different responses, just like the different receptors can. So it matters what those receptors are and what they're coupled to. And then downstream, the type of cell matters, because if it's a muscle cell, it's going to have different structure than a nerve cell is, and so you're going to get a different response from that. There's also reasons why you might have different responses to the same hormone in the same cell at different times. So upregulation is when you have more of a receptor present on the cell than was originally there. So there's a number of different reasons why you can have upregulation of a receptor, but that puts more of the receptors on the cell surface, giving more of a chance of the hormone to bind. You can also have downregulation where you remove the receptor. So if you remove the receptor, obviously its presence is gone, and so that's going to be an issue as it can no longer bind. A cellular response can have several different types of effects. And one thing to keep in mind is that how a cell responds may depend on the presence of more than one hormone. So each cell is going to have multiple receptors on it. The different receptors that are present may lead to different responses depending on what hormone's present, and the combination of those hormones can also matter. So here we describe a permissive effect where binding of the hormone to one receptor leads to no effect. Binding of a different hormone to another receptor leads to no effect. But when you have both hormones bind to both of the or to their respective receptors on the same cell, you get a target cell's response. So this is essentially allowing where one hormone allows another to interact. An example of this is similar to how iodine deficiency can cause a lack of thyroid hormones to be produced. Those thyroid hormones are necessary for the reproductive hormone to have their actual effect on the development of the reproductive system. So an iodine deficiency can lead to delayed development because you don't have the the ability for those hormones to actually produce their effect. Another effect you can have is a synergistic effect. This is where one hormone has a positive effect, a stimulating effect on the target cell. The other hormone also has that stimulating effect. And then when you have a combination of those, you get an amplified response. An example of this would be the follicle stimulating hormone from the pituitary gland and the estrogens from the ovaries. Both of these stimulate the ova, but you need both of them to get the adequate response for the maturation of the ova. Finally, you can have what is known as an antagonistic effect. This is where one hormone stimulates its receptor, leading to a response, but the other hormone leads to an inhibitory response. So the two hormones acting together can cancel each other out. So this happens when two hormones have an opposite effect. So if glucagon and insulin were both binding to the cell, because they have opposite effects on the cell, that is going to essentially cancel each other out. The endocrine glands themselves need to be stimulated in order to know when to release their hormones. The release of the hormones can come from stimuli. There are three major types of stimuli. So one is humoral stimuli. So humor, think blood, so changes in the levels of non-hormone chemicals, things like ions or nutrients. This is what the body is sensing and is causing the release or the inhibition of the release of a hormone to help maintain homeostasis. Osmolarity is a good example of this, so it regulates the release of ADH by the hypothalamus. Blood glucose is another example of this. It regulates the release of insulin and glucagon by the pancreas, depending on whether blood glucose levels are high or low. Another type of stimuli is neural stimuli. So this is where stimulation of the endocrine gland comes from neural input. An example of this would be the sympathetic nervous system stimulating the adrenal medulla to release the catecholamines, norepinephrine and epinephrine in response to stress. So this is triggering the fight or flight response. You are having a signal here that needs to communicate, hey, we need to get these hormones out quickly, and so the neural stimuli helps the release be very rapid in this case. And then finally, you can have a hormonal stimuli. This is where the stimulation of one endocrine gland by another hormone may result in the secretion of those of another hormone. So you have a stimulating releasing hormone that's produced in another gland. Hypothalamus is a classic example of this. It regulates the secretion of many hormones in the anterior pituitary, which in turn stimulate other hormones. A term to describe the stimulating releasing hormones are tropic hormones. And so if you hear tropic in the name of the hormone, that generally indicates that it's a hormone that stimulates another endocrine organ to release their hormone. So one thing to note is that the stimulus can be modified by the nervous system. So the stimuli and the turn-off factors can be regulated by these nervous system. So without this added safeguard, the endocrine system would be very mechanical in nature, and it would be very much like a household thermostat. But the issue with this is that the thermostat can't accommodate itself when changes need to occur. You have to go in there. So imagine you're going on vacation. You still don't want your house to get super hot, but you don't need the air conditioning on like when you're present. So you can go there and change it yourself. That's sort of like you acting like the nervous system to modify this. So an example of this is the blood glucose levels. Normally, they're regulated by to be around 90 to 110 milligrams per deciliter. If you have higher or lower, you have a response that shifts this. But during stress, the neuromodulation can modify this so that you have higher levels. And so this basically raises the set point that you're at. And so you still have regulation occurring, but it's going to be triggering a effect that's adjusting the way that the energy is being used. This is sort of a reminder about homeostasis and what homeostasis is doing. So homeostasis means similar standing. So it emphasizes not a static but a dynamic response, and it helps maintain stable internal conditions. You have a stimulus, which is what produces a change in the variable. You have a receptor that detects that change. You have the afferent pathway that carries the information to the control center, which monitors the set point. The set point is going to if it reaches or if it goes out of that set point, it's going to take a message to an afferent pathway that carries it to a effector. The effector is what's reacting and returns the body back to what it was, which is what its response. So this is the driving thing that the endocrine system is really doing throughout the body. So let's look at an example of this as it relates to the endocrine system. So you have hypothalamus glucocorticoid sensors in the blood vessels. These are going to monitor the presence of the glucocorticoids. So the release of the glucocorticoids, which are coming from the adrenal system, it's being triggered from hormones from the hypothalamus and pituitary gland. And so the signal is inhibited when glucocorticoid levels are elevated and are going to be responsive to it. So glucocorticoids, one that you're probably familiar with is cortisol. So we have an imbalance occurring with our glucocorticoids. This is going to lead to the hormone release by the hypothalamus. So it's going to release the corticotropin. There's that tropin term to tell you that it's acting on another endocrine gland. So the corticotropin releasing hormone is going to be released from the hypothalamus. It triggers a hormone cascade by signaling to hormones in the anterior pituitary to release the glucocorticoid into the blood. So CRH leads to glucocorticoid release, which is then going to increase the amount of glucocorticoids in the blood. So this is the corrective response. As these increase, you have the negative feedback loop occurring. So this inhibits the corticotropin releasing hormone release. The hypothalamus is perceiving this change, and so it stops releasing this, and then homeostasis is maintained. And it stays in homeostasis until another imbalance is reached one way or another. So that was an overview of the endocrine system. In our next lecture, we'll talk about specific endocrine organs, the hormones that they release, and what the effects of those hormones are.